Hi, everybody. So today we're going to be talking about photosynthesis. So we're going to look at the entire thing. Um, so photosynthesis has been around for a long, long time. We'll focus on how plants capture light. All right. So this ability of plants, right, to capture sunlight and store its radiant energy in the form of sugar, it's been around for geez, probably 3.4 billion years. It evolved way before multicellularity. It likely evolved before even eukaryotes evolved. What's interesting is that probably every organism that photosynthesizes today is probably descended from these ancient ancestors. And so what they probably were is they probably were a cyanobacteria, or these purple sulfur bacteria are a completely unique strain too that also capture the radiant energy from the sun. And then eventually they components of each one of these stuck around in the eukaryotes once the domain of eukaryota evolved, and then eventually they integrated into things like multicellular protists like this algae, or unicellular eukaryotes like these euglenas, and then or into like the plants that you're familiar with today, like multicellular plants. So anyways, all of these organisms share the same food structure, meaning they are autotrophic, meaning auto like automobile, it moves itself. Autotrophic, so auto food gathering, like it's automatically gathers its own food from the sun. Okay, so let's take a look at the, whoa, hello. Let's take a look at the anatomy of a plant leaf. So if we take a look at the plant leaf and we kind of cut a cross section of its, let me get my pointer out. If I take a cross section of the leaf, right, and I look at the top side of the leaf and the bottom side of the leaf, and then I've got this cross section, what I can see is that there's a lot of these like palisade cells that have these organelles called chloroplasts inside of them. Now these palisade cells, if I was to zoom in and take a look at some of the chloroplasts that are inside of them, right? This is the palisade cell, right? Which is part of the mesophyll, meaning middle layer of the plant, right? Meso, middle. The palisade cells are in the mesophyll, and the palisade cells have these, are just packed full of chloroplasts because the palisade cells are primarily in the leaf where photosynthesis is taking place. And the chloroplast is the organelle of photosynthesis. And so you've got cross section of plant leaf, right? Mesophyll, palisade cell packed full of these organelles called chloroplasts. Now, another part of the anatomy we need to look at are these tiny gate-like structures called stomata, or stomates. So what these stomata are, is they're tiny pores in the surface of a leaf, and all of the other junctions between the cells are sealed tight, and usually they're kind of waxy too, so they're watertight. But the problem is that if plants are taking in carbon dioxide and making sugar, and the byproduct of photosynthesis is oxygen, well, they're going to end up with an abundance of oxygen inside of the plants and a shortage of carbon dioxide inside the plant. And so these gate-like structures allow for the transpiration of gases like carbon dioxide and oxygen. So oxygen, because it's in higher concentration inside the cell, will diffuse out through the stomata, and then the carbon dioxide, because it's in higher concentration outside of the plant leaf, will spontaneously diffuse into the plant leaf and into the chloroplasts. Okay, so stomata, those are those structures. Now there's one more molecule that the plant is primarily concerned with losing. The reason that these structures don't stay open all day long, the reason that they're gates and they regulate when they open and close, is because of this right here, is water. The water molecule will diffuse and will spontaneously evaporate and transpire out of the stomata. So in the desert, right, if you look at the stomata on desert plants, they only open at night when the sun's not shining to allow for the transpiration of gases. So they limit the amount of water that they lose. Okay, so we'll take another look at the anatomy of a chloroplast, right? So you have an actual electron microscope, a colored electron microscope diagram here, whereas you have like kind of an artistic depiction up here. So there's two outer layers of membrane, right, between the chloroplast and the cytoplasm. But inside of that inner membrane, right, that's where I want to focus on. And that's where we have these structures, thylakoids, right? So if you'll notice, right, there's a little like bracket here that's saying, hey, look at one of these coin-like structures. That represents a thylakoid. And then if you look, right, a whole stack of these endomembrane structures, a whole bunch of them stacked on top of each other makes a granum. And so thylakoids are stacked on top of each other to make granum. And then they're surrounded by this liquid in the middle of the cell, or sorry, in the middle of the chloroplast called the stroma. And so those are the three terms I want you to know. The thylakoid are the little disc-like structures that have the photosystems in their membrane, like the light harvesting complexes. The stack is the granum, and then the dense liquid inside the chloroplast is the stroma. All right, so let's take a look at photosynthesis, like the equation itself. So remember, right, from cellular respiration, all 6 carbons from glucose get turned into carbon dioxide. And then if you think about it, right, all of the electrons from glucose, right, once they get ripped off of the glucose molecules, those electrons eventually get put into cellular batteries like NADH and FADH2. And then those cellular batteries drop those electrons off on the electron transport chain, and they travel down the transport chain, pumping protons into the inner membrane space. When they get to the end of the electron transport chain, they join up with oxygen, like the air you breathe, right? They join up with oxygen, they join up with the electrons and then some protons, and they make water. So you've got, these are the 6 carbons from glucose. These are the waters made from the electron transport chain. And then photosynthesis takes the byproduct of our cellular respiration and turns it into sugar, oxygen, and more water. And so we'll see where all 3 of these come from in the process of photosynthesis. Yeah. But we can ask this question, right? So if we're looking at energy, right? If we're to look at this side of the equation, right, before the arrow, and look at this side of the equation after the arrow, right? We have reactants over here and products over here. And so this is what we finish with, and this is what we start with. And remember, when we're trying to figure out delta G, we take the free energy that we end with and subtract that from the free energy we start with. So the question is, which one has more free energy? Does the products with glucose, oxygen, and water have more free energy than carbon dioxide and water? Think about where carbons are bonded to hydrogens and the proximity of carbons to hydrogen. Look over here, there's no hydrogens attached to this carbon. Look over here, there are hydrogens attached to that carbon. So which side has more free energy? This side. So you take what you finish with, subtract it from what you start with, and what we find out is that the delta G is positive, right? You end up with more than you started with. You end up with more free energy in your system than you started with. So what does that mean? Delta G will be positive. So wait a second, that means something is missing from this equation. Energy, right? Energy has to come from an outside source. This is an endergonic reaction, right? This is a anabolic reaction. This is a building reaction. We're adding energy into our system here, right? And it's reflected in our sugar molecule. Okay, so we're going to look at the energy that comes from the sun in more detail, and we'll look at it right here. So what this is this is your electromagnetic spectrum, okay? And this is done on a logarithmic scale, and it's done basically with, it's using nanometers as a unit of measure. So a meter is like, oh geez, a meter is, 3 feet long. 1/1000th of a meter is a micrometer, and 1/1000th of a micrometer is a micrometer, and then 1/1000th of a micrometer is a nanometer. So nanometers are really small, right? But if we're measuring wavelengths on a invisible photon, right, then it doesn't matter. They're going to be small wavelengths. So, sorry, I digress. Regardless, electromagnetic radiation is the radiation of photon energy. Now, the weird thing about photons is that even though they have 0 mass and they travel the speed of light, they can behave like a particle because they can be absorbed by electrons. So electrons can absorb photons and electrons can give off photons. And photons, remember, are this weird, photons are so weird. They're weightless. They don't have any mass. They behave like a wave, so there's a wave function attached to them. So you have a distance between the peaks and the troughs, right? So just like waves in the ocean, right? What's the distance between this wave and that wave? That's the wavelength, okay? So when we're talking about electromagnetic radiation or photons, we're saying what's the distance between the peaks in that photon wave? And so the distance between peaks in that photon waves are what give that photon its identity or its characteristics. So if you have really long peaks in between the waves, that's going to be things like radio waves, right? So even when you tune in your car radio, right, you're picking up on invisible electromagnetic radiation. You can't see it. It's not part of the visible light spectrum, but it is electromagnetic radiation. And the peaks in between, the distance between peaks is huge. It's enormous. It's like, oh, what's 10 to the third? Like that's 1,000 meters in between peaks. I mean, that's so high. It's hard to even fathom what that is, right? Microwaves, like another large number distance between peaks and troughs. Infrared light, that's what photons of warmth, right? So when you infrared, right? So that's where you can't see it, but sometimes like you can have infrared like optics on like scopes on guns and stuff, and it picks up on wavelengths of light that are a little bit longer than the visible spectrum. So infrared night vision goggles and stuff like that pick up on a little bit more of the longer end of the electromagnetic spectrum. And then you get your visible light spectrum, right? And so this is stuff that we can see. Whenever you see something that's reflected at you that looks red in color, you know that the photons that are striking the nerve receptors in your eyeball, right? The photons are around 7 to 750 nanometers long, the wavelengths are. And then as the distance between the wavelengths decreases, so as it gets shorter and shorter and shorter, we see those photons as yellows, greens, shorter and shorter, blues and purples. And then suddenly they get so short that they don't actually stimulate the nerve receptor in your eyeball, and those become ultraviolet light. And so it's not visible, right? But it's still electromagnetic radiation. And then let's say you break a bone, you need to go to the doctor. He's going to shoot electromagnetic radiation at your body that goes through your cells, right? It won't go through bone, but it'll go through your tissue. And so it'll show you where the broken bones are in kind of like a shadow way, like your bones create a shadow imprint on the film. And then you've got gamma rays, and they're so short. They usually are vibrating up and down so fast. They don't even slow down when they hit stuff. They just go right through it. So you got gamma rays going through our planet right now. So anyways, that's the spectrum of electromagnetic radiation. The ones we're concerned with, right, is this visible light spectrum. Okay. So visible lights, all EMR is coming from the sun, right? So all wavelengths is coming from the sun. The sun is giving off radio waves, microwaves, gamma rays, X-ray. It's a big giant ball of radiation in the sky. And so only portion of that spectrum is being absorbed by the chloroplast, right? The absorbed light is going to be the reds, the oranges, the yellows, the blues, and the purples, but the green just goes right through it or is reflected. And so green light does not interact with any of the molecules or pigments inside of the chloroplast. Green light gets reflected or green light gets transmitted through it. It doesn't even get absorbed. And so the reason we know this is we can take something like white light and we can prism it, and we can use, when we say we prism white light, that means we're going to use just one wavelength of light and shine it through a sample and see how much of that wavelength is absorbed and how much of it is transmitted through the substance, right? Or vice versa. So how much is transmitted through, right? Or how much is absorbed? It depends on how you flip the equipment, right? How you set it up to read. Okay. So green light, if we shine prismed light through the sample, right, the green light will make it all the way through to the other side, and the photoreceptors on the other side will say, oh yeah, I'm getting 100% of the light of the green light I'm shining through is being transmitted through the sample of chlorophyll, right, a pigment in the chloroplast. And then take something like blue light, and blue light is absorbed by the pigments in the chloroplast. The blue light is, the green light isn't, but the blue light is, and it says, okay, there's not a lot of being transmitted through the sample. The sample is absorbing a lot. And so what we can do is we can actually plot this out on a spectrum, and we can say, okay, if I shine 400 nanometers, right, through this sample, right, how much of that nanometers, that wavelength of light is being absorbed by my chlorophyll sample? And so if I'm able to separate all of the different pigments inside of the chloroplast, right, the chlorophyll A is one type of pigment that absorbs in sort of the dark blue, light purple spectrum, and also towards more of the reddish spectrum. If you take chlorophyll B, that's another pigment that picks up light probably around the bluish spectrum, and then also a little bit around the orange spectrum. And then you have carotenoids, another pigment, right? Another protein molecule pigment that's going to pick up on kind of the bluish, the light blue light, and then hardly any of that is actually going to get picked up in the upper, the longer wavelengths of that spectrum. And so what this shows us is it shows us that like no green light is actually used or absorbed by the pigments in the plant, which is what makes them green. All right, so this is a porphyrin ring. It's a light absorbing molecule. Something that's interesting, right, is so this is sort of like your photoreceptor, if you will. This is like your solar panel that when the photon strikes it, it charges up some of the electrons in this solar panel, right? And this solar panel is embedded into the thylakoid membrane, which is a phospholipid bilayer. And this hydrocarbon tail is sort of like the anchor. It just sticks into the membrane because like dissolves like, and it can stick into the membrane. And then this photoreceiving head sticks out of it. And so this particular one is chlorophyll A, right? Pigment. And then it's a CHO, it's an aldehyde in chlorophyll B. And that changes what wavelengths of light are absorbed by this like solar panel up here. Okay. So anyway, something to think about, right? So we can look at it like this, right? So there's a fluorescence to electrons. So a photon comes in, right? A photon of light, a wavelength of light comes in and strikes the pigment. The pigment absorbs its energy and transfers that energy into an electron. So that electron becomes into an excited state. That electron is going to give off some entropy, right? Because this electron now has some free energy because it absorbed the free energy of the photon, and now it's up here, and it's like, whee! And so that electron will give off some of that free energy and transform it into entropic energy, right? That's the heat. And so that lowers its free energy state. It will also give off photon fluorescence. So like it'll actually glow in the dark. This is how glow in the dark substances work, is the glow in the dark substance catches the light, right? It charges up the electrons in the substance, and then you turn off the lights and you can see it fluoresce. You can see the glow in the dark substance give off photons because it glows. It's got light, right? And so this is the idea of a chlorophyll molecule, is that it can absorb light, charge up electrons, and that's all that's important for this. Okay, let's take a look at the photosystems that are inside of the plant membrane. I know this is a complicated drawing. I usually draw this pretty simply on the whiteboard, but this will suffice for what we're talking about. Okay, there's two photosystems that exist. So two sets of solar panels that exist inside of the thylakoid membrane. Notice they're a transmembrane complex, so part of it's sticking out into the stroma and part of it is sticking into the thylakoid space. Okay, so let's start with step one. Step one, light's going to come in and strike this solar panel here called photosystem 2. Photosystem 2 is then going to take water, so this is why plants need water. It's going to take water and it's going to strip the electrons away from it. Well, what that does is that makes oxygen gas as a byproduct, and that oxygen gas is just going to diffuse out. And so that's why plants are able to make oxygen, is that they take the electrons from water, right? The protons float off into space, and we'll deal with those in a second. And then the oxygen that's left over diffuses as gas out of the cell. And so if you think about it, right, like photosystem 2, absent of the light that's like striking it and charging it up, photosystem 2 is a lot like subunit 4 of the electron transport chain, just in reverse, right? Because remember, subunit 4 collects the electrons that flow down it. Oxygen is sitting at the end, and those electrons join oxygen with a couple of protons and become water. So this is the opposite of subunit 4. So anyways, the water molecule is split. The electrons are stolen from water, right? The oxygen gas diffuses out, and then the protons are left inside of the middle of the thylakoid here. Okay, those electrons are excited, right? They're transferred to the chlorophyll, and the electrons are excited by the light. So the light comes in from the sun, it excites the electrons. They're like, whoa, boy. Then those electrons, just like they did in the electron transport chain, are transported through a series of subunits that pump protons into the thylakoid. So this is opposite of the mitochondria. Remember, mitochondria, they're pumping it out of the middle into the intramembrane space. In the chloroplast, they're pumping it from the intermembrane space, so to speak, or the stroma, back into the middle. And so protons are building up in the middle of the thylakoid as the electrons are being passed from subunit to subunit. So when they get to photosystem 1, the other solar panel, right, they've lost a little bit of energy. Those electrons were used to fuel some pumps to pump protons in here. And so that use of its energy drops its energy state, and it needs to be charged up again. So the light, the light from the sun is going to hit the solar panels, right, the chlorophyll in photosystem 1. It's going to transfer those energies to the electrons. They get excited, and then photosystem 1 passes the electrons to this enzyme complex called NADP reductase. And so NADP is just nicotinamide adenine dinucleotide phosphate. And the P, for me, I just remember it's stored in plants only, plants, P, right? So NADP reductase is going to take those electrons that were originally from water that powered all of these proton pumps, right? It's going to take those electrons that just got charged up in photosystem 1 and transfer them to NADP, reducing it, right? So anytime we take electrons from somewhere and put it on something, the thing that's gaining electrons gets reduced, right? And so remember again, right, that hydrogen, that proton comes in and corks the cellular battery, so to speak, right? So the electrons pour into NADP plus, right? They pour into it, and then the hydrogen proton caps it. And so now this NADPH is like a charged up battery that can go enter a cycle and deliver its energy to a sugar molecule and to make a sugar. Okay, now remember, let's zoom in a little bit, right? So that was, oh, hello. So let's deal with these protons too. Okay, so we've got the light harvesting complexes, right? Photosystem 2, photosystem 1. They're solar panels, right? They're capturing electrons from water, charging them up. The electrons are powering these proton pumps that pump protons into this space in the middle here. And then just like in the mitochondria, right? Those protons are going to build up in concentration, and they all have the same charge, right? So it's like the same end of a magnet all packed in there together. Molecules are magnets. And so they're pushing against each other, repelling each other. All they want to do is escape and find equilibrium. Well, look, ATP synthase, the same exact enzyme complex is in the mitochondria. ATP synthase gives them that water wheel exit where they can flow through ATP synthase, and that flowing of protons out of the thylakoid is what provides the rotational energy, the torque, if you will, to compress an adenosine diphosphate and an inorganic phosphate together to make ATP. And so this whole process, right, if we whittle it down to what's really happening, electrons from water are getting stripped away, charged up, and placed into cellular batteries, right? Placed into NADPH. So electrons from water get charged up by the sun, right? They end up in NADPH. The protons are going to flow through ATP synthase and make ATP. So the two energetic byproducts of the light reactions are NADPH and ATP, and they're going to go enter the Calvin cycle that takes place in the stroma. All right, so this is just a zoomed in image of what we just saw. So remember, right, ATP synthase is going to use protons to make ATP, right? The flowing of protons from inside the thylakoid to out. And then the photosystem 1 is going to transfer its charged up electrons to NADP reductase to make NADPH. So now you've got a reduced cellular battery here, a reduced cellular battery here, and those can power the dark reactions. Okay, one thing I'd like to point out, right, the only difference between ATP synthase in the mitochondria and the one in the thylakoid is that they're oriented in opposite directions, right? Mitochondria, the protons are building up in the outside and flowing in. In the thylakoid, they're building up on the inside and flowing out. I have a strong suspicion that they share a genetic history with each other billions of years ago, that they evolved from a similar process, and each one specialized in something else, right? So it's because there's a lot of similarities between them, right? Between the electron transport chain and the mitochondria and the light harvesting complexes, the photosystems and the thylakoid. They're very similar in terms of function. They're almost like inverted, if you will. It's kind of neat. Okay, the dark reaction. So how do we take the energy from ATP and NADPH that we got from the light reactions, and then how do we use that energy to reduce carbon dioxide into sugar, right? So we're trying to take carbon dioxide and make sugar. So anyways, this is just a scaled down version of what's happening, right, on a general level. Water comes in, right, to the system, into the light reactions, into the thylakoid. Water comes in, its electrons are charged up by light, by sunlight. Those electrons are transferred to NADP and ADP and inorganic phosphate, which makes, go back, right? Which makes ATP as a byproduct and NADPH. So these cellular batteries get charged up from the electrons from water that get charged up by light. So these cellular batteries are capturing the electrons from water that have been charged up by the sunlight. The byproduct of all that, right? Once you strip away electrons from water, is you get oxygen gas as a byproduct. And that's what we use to breathe. And that's what fuels our mitochondria. It's crazy. Okay. So anyways, the charged up batteries then are going to enter this Calvin cycle, which takes carbon dioxide out of the atmosphere, puts a bunch of electrons on it. So remember, if we take carbon dioxide and we reduce it, we can turn it into a sugar. And so that's what the Calvin cycle is going to do. It's going to make a sugar molecule. All right. So how to make sugar? How are we going to do it? We need carbon. Where do we get it? We're going to get carbon from carbon dioxide. Is the reaction endergonic or exergonic? Whew, good question, right? So let's think about it, right? Sugar is the end state, right? Carbon dioxide is the beginning state. Which one has more free energy? Sugar. Take the final state minus the initial state. So you have more at the end minus what you had at the beginning, which is less. You should end up with a positive change in free energy, which makes it endergonic. So what needs to come in? Well, energy from the sun, right? So we got to get energy. And where does it come from? It comes from the sun, but remember that energy is also coming from those cellular batteries we charged up, right? So the energy indirectly, the energy that makes sugar indirectly comes from the sun, and it's transported to the Calvin cycle by these cellular batteries, right? All right, so this is how the Calvin cycle starts, right? We start with this molecule called rubisco bisphosphate, 1,5-bisphosphate. All right, so if you'll notice, pay attention to where we have 3 of these 5 carbon molecules, okay? And maybe I can annotate. No, I can't, because if I skip the slide, it won't let me annotate. Okay, forget that. All right, so we have 3 5-carbon molecules. Let's keep track of the carbons, okay? So that means there's 15 carbons here. We're going to take 3 carbon dioxides and we're going to attach them to those 3 molecules. So that's 1 apiece, right? So you have a 5 carbon here, and then we're adding a carbon dioxide to it, right? So now we have 3 6 carbon molecules. Now watch. We can cut those 3 6 carbon molecules right here, cut them right in half. And then we have 6 3 carbon molecules. Those 6 3 carbon molecules are going to grab some of the phosphates from ATP, right? So remember, that's adding energy into the system. So now you've got 6 3 carbons with 2 phosphates attached, right? So a highly energetic molecule here. Then we're going to add some electrons to that molecule from our NADPH. So we got a bunch of NADPHs come in, right? They drop off their electrons onto this molecule, and then a couple of the phosphates pop off as we add on the electrons. And so now we've ended up as G3P. So we've reduced 1,3-bisphosphoglycerate. Think about this. I know I didn't ask you guys to memorize it, but this molecule right here is in glycolysis. So this is almost like reverse glycolysis in a way, in a weird sort of way, right? So we have 1,3-bisphosphoglycerate, right? We go backwards. This is that step in glycolysis where we go from G3P once we split fructose in half and we go to 1,3-bisphosphoglycerate, right? That big drop in free energy step I talked about. That's this in reverse, right? So now we're actually going in reverse. We're taking the electrons from NADPH and putting them into the system now. So it's endergonic. We're building up some G3Ps. All right, so now, right, we're counting the carbons. We've got 6 3 carbons with 2 phosphates attached. Now we have 6 3 carbons with only 1 phosphate attached because some of them popped off, right, to make room for the extra electrons. So now we've got 6 G3P. 1 G3P is going to leave the system, and we'll talk about what happens to that G3P, right? If we have 2 of these G3Ps, right, we can use that that isomerase, and we can isomerize it into DHAP, like dihydroxyacetone phosphate, and then join those 2 together and make fructose 1,6-bisphosphate, isomerize that into glucose 6-phosphate, and then remove the phosphate with a kinase, and then now you have glucose. So we can actually take this molecule and build a sugar out of it. The remaining 5, so we had 6 G3P here. We lost 1 of them, so now we have 5 G3Ps. So count the carbons, right? That's 5 3 carbons, so that's 15 carbons. right? We want to regenerate our rubisco bisphosphate, 1,5-bisphosphate, our RuBP. We want to regenerate it. So we're going to use the energy of ATP to rearrange it, to isomerize it, right? To rearrange them and turn them back into rubisco 1,5-bisphosphate. So we went from 5 3-carbon molecules with some energy from ATP, rearrange them into 3 5-carbon molecules, and then we start the cycle again. So 3 carbon dioxides yield 1 G3P. So look, that's 3 highly oxidized carbons now yields 1 3-carbon sugar that's highly reduced. So low free energy, we're adding a bunch of free energy to it, and it becomes a sugar, or half a sugar, if you will, right? So what do we do with glyceraldehyde 3-phosphate, right? So the plant is gonna ask 2 questions. The plant's gonna ask, do I need immediate energy? Do I need to get more ATP to do some sort of like building in the cell, right? If that's the case, right, G3P is a byproduct of glycolysis. It's one of the intermediate steps. So the plant can just literally transport G3P straight out into the cytoplasm. So it crosses the chloroplast membrane. It crosses the membrane, goes into the cytoplasm, and then it enters glycolysis at that step and goes through it and goes through aerobic respiration. Because remember, plants have a mitochondria too. So they will go through, they'll take G3P, right? They'll oxidize it to pyruvate, and then they'll send pyruvate to the mitochondria inside the plant. Now, if the plant is not in a state of like growth, right, or it's not making fruit or something, it might store that G3P as a starch. So remember, G3P will have to be isomerized into dihydroxyacetone phosphate, and then it'll be joined to another G3P, which will make fructose 1,6-bisphosphate. They'll remove a couple of the phosphate groups, turn it back into glucose. The glucose will then get dehydrated and turn into a polymer starch like amylose, a complex carbohydrate. I know that's like, what? It's a lot, right? I'd get it. It is a lot. Okay. So if you need energy, you send it to the cytoplasm for glycolysis and cell respiration. If you need to store that energy for later, you can send it out to the amyloplast and it'll turn it into a starch and store it. Or it might turn it into part of the cell wall, right? Like a cellulose structure. Okay, and that is it for photosynthesis. Hope you guys enjoyed it.